Volvox occurrence. Volvox is a colonial alga. It grows in fresh water of pools, ponds, etc. It is represented by about 20 species. Single colony looks like a small ball about 0.5 mm in diameter. In rainy season the color of the ponds becomes greenish due to rapid growth of Volvox. Plant body and cell structure. Plant body of Volvox is a cenobium, like a hollow sphere of gelatinous substance. In the hollow sphere, huge number of cells are arranged towards periphery in a single layer. The number of cell varies from species to species and it ranges from 500–60,000. The cells are spherical in shape having cup-shaped chloroplast, with one or more pyrenoid, an eye spot, 2 to 6 contractile vacuoles and a single nucleus. Each cell has 2 equal flagella placed anteriorly. Adjacent cells remain connected by cytoplasmic strands formed during cell division. The central region of the cenobium is generally hollow but in some cases it is filled with gelatinous material or water. The cells of the anterior region have large eye spots than the posterior region, indicating the clear polarity in the cenobium. Important features of Volvox. Plant body is cenobium and consists of large number of biflagellate, pear-shaped cells. The cells of the cenobium are connected together by means of protoplasmic strands. Young cenobia consist of only vegetative cells and are concerned with locomotion and food production while older cenobium consists of vegetative cells, daughter cenobia and antherozoid mother cells and/or ovum mother cells. Sexual reproduction is oogamous and the cenobia may be monoecious or dioecious. The female gametes or ova are large and non-motile, produced singly inside the oogonium. The male gametes or sperms are spindle-shaped, narrow with a pair of apical cilia and are produced in bunch inside the Anthridium. The result of sexual union is the zygote, which on germination develop into a new cenobium either directly or by the formation of single biflagellate zoospore. Reproduction of Volvox. Volvox reproduces both asexually and sexually. Asexual reproduction takes place during favorable condition, but the sexual reproduction occurs during unfavorable condition, i.e., towards the end of the summer months. Asexual reproduction. A few cells at the posterior side of the cenobium enlarge about 10 times. The cells withdraw their flagella and become more or less round. They are pushed inside the colony during their development. These cells are called gonidia or parthenogonidia or autocolony initials. The gonidium is separated from the vegetative cells by its position and size. Sexual reproduction. Volvox reproduces sexually during unfavorable condition, i.e., towards the end of growing season, late summer. The sexual reproduction is oogamous. Species may be monoecious or dioecious. Most of the monoecious species are of protandrous type, i.e., Anthridia develop and mature earlier than oogonium. Some cells of the posterior region of the colony withdraw their flagella and develop into reproductive bodies called gametangia. The male gametangia are called Anthridia and the female as oogonia. Development of Anthridium. During development of Anthridium, an anthridial initial becomes differentiated from the colony. It is like a gonidium, which is larger in size than vegetative cells and contains dense cytoplasm with a single nucleus. The cell undergoes repeated longitudinal divisions like the asexual stage and forms generally about 64 to 128 cells, though the number varies from 16 to 512, depending on species. Like the asexual stage, the cells are arranged in groups and then undergo inversion by which the anterior side of the cells faces towards outer side. Each cell develops into unicellular, elongated, fusiform, naked and biflagellate antherozoid. The antherozoids are released individually. In some species they are also released in groups. Development of oogonium. Single vegetative cell of the colony at the posterior side withdraws its flagella, enlarges in size and become a more or less flask-shaped oogonium. The entire protoplast, without undergoing any division, forms a uninucleate non-flagellated egg or female gametophyte. The egg or female gametophyte is spherical, uninucleate, non-flagellated, green in color and has parietal chloroplast. It has many pyrenoids and large amount of reserve food. The mouth of the flask-shaped oogonium opens towards the outer surface of the colony. Fertilization. After maturation, the antherozoids, spermatozoids, are liberated from the antheridium either singly or in mass. They move in water and get attracted by the chemotactic stimulation to the surface of the eugonium. A few antherozoids enter near the egg by breaking the eugonial wall with the help of proteolytic enzyme probably secreted by the antherozoids. Out of many antherozoids, only one entered into the eugonium. Only one succeeds to fertilize the egg and forms a zygote. Oozygote. The zygote secretes a thick wall around itself. It accumulates the hematochrome and becomes red in color. The wall of the zygote may be smoothly or spiny. Zygote is liberated by the disintegration of the mother wall and remains dormant for a long period. Germination of zygote. During favorable condition, the zygote germinates. Before germination, the diploid 2-end nucleus of the zygote undergoes meiotic division and forms 4 haploid cells.